Good morning. It's September 15th, and this is your daily brief in science. Here's everything you need to know. Bangladesh's Globe Biotech has secured a U.S. patent for its COVID-19 vaccine, Bangavax, marking the first international patent for a vaccine from Bangladesh's pharmaceutical sector. The vaccine's technology was published in Nature and recognized by WHO in 2020. It can be adapted for multiple platforms, including DNA, protein, inactivated and recombinant virus, with potential applications for cancer, diabetes, and autoimmune diseases. The announcement was made during a press conference at Globe Pharmaceuticals' headquarters in Dhaka's Tejgaon industrial area. The patent was officially granted on August 26, 2025, after applying on September 14, 2021, and is based on a priority claim under the Paris Convention. Key figures at the event included scientists and executives from Globe Biotech, Globe Pharmaceuticals, and Delta Pharma. Globe Biotech leadership cited weak patent laws in Bangladesh as a motivation for seeking the U.S. patent to protect their innovations and support global expansion. The company highlighted potential vaccine exports could generate foreign exchange and earn international respect, with interest from 23 countries, including ASEAN nations. They estimate that with government support, they could have secured orders worth $5 billion during the pandemic, reflecting strong international interest. Despite this progress, the company experienced delays in regulatory approval and human trial permissions, which they attributed to issues with government support. Globe Biotech views the patent as an acknowledgement of Bangladesh's technological capabilities and expects final testing for public availability to begin soon. Recent research into plant stem cell regulation could transform crop development by improving plant regeneration, boosting biodiversity, and creating more resilient and productive crops. Plant stem cells are essential for maintaining food supplies, livestock feed, and fuel sources, yet many of the genes controlling these cells are still largely unknown. Understanding these regulatory mechanisms may lead to better techniques for plant breeding and regeneration, advancing knowledge of plant diversity and resilience. Researchers at Cold Spring Harbor Laboratory mapped key stem cell regulators like clavida-3 and wuschel across thousands of maize and Arabidopsis shoot cells for the first time. They used single-cell RNA sequencing to create an open-access gene expression atlas of around 6,000 cells, providing a valuable resource for plant biologists. This foundational data identified hundreds of genes that are preferentially expressed in plant stem cells, emphasizing their evolutionary importance across species. The goal is to support scientists and breeders in developing crops tailored for food, fuel, or feed, especially amid climate change and global food security concerns. The findings, published in Developmental Cell, serve as a key resource for improving crop yields and traits. The Gene Expression Atlas enables researchers to access this data without repeating experiments, accelerating progress in crop science. An international team has achieved a breakthrough in black hole research by measuring the speed and direction of a black hole's recoil after a merger. This was accomplished using gravitational wave data from the 2019 event GW190412, which involved two black holes of significantly different masses. The analysis focused on the kick received by the black hole, causing it to move rapidly away from its original location. This method, developed in 2018 and confirmed with detectors like LIGO and Virgo, analyzed differences in gravitational wave signals based on the observer's position. By doing so, scientists reconstructed the black hole's full 3D motion, demonstrating advanced capabilities in gravitational wave astronomy. The event, GW190412, provided a long-duration signal suitable for detailed analysis due to the disparity in black hole masses, approximately 8.4 and 29.7 solar masses. Understanding the recoil velocity helps distinguish genuine signals from background noise, especially in dense environments like galaxy cores. The measured recoil speed is about 150 times the speed of sound on Earth, enough for the black hole to escape its dense stellar environment. Although the exact location remains uncertain due to the 2.4 billion light-year distance, data suggests the black hole may be traveling out of its original environment, such as a globular cluster. Future research aims to combine gravitational wave recoil measurements with electromagnetic signals to gain further insights. This development marks a significant step forward in understanding black hole dynamics and evolution, expanding the capabilities of gravitational wave astronomy. Lion TCR has received FDA IND clearance to begin Phase 1b/2 clinical trials for its innovative TCR T-cell therapy, Lyo6M004, targeting chronic hepatitis B. This makes it the first therapy of its kind to enter clinical development for this disease. Developed by Lion TCR, a company originating from Singapore's A-Star, the therapy is the first HBV-specific TCR-T treatment to gain FDA approval for a Phase 2 trial internationally. It aims to address a global patient population of over 290 million people living with hepatitis B. The company's in vivo TCR-T platform offers advantages such as shorter production times, lower costs, and increased accessibility compared to traditional ex vivo T-cell therapies. Validation from a major U.S. cancer research institute supports the therapy's efficacy, demonstrating significant effectiveness in animal models. Lion TCR uses AI discovery and advanced RNA technology to develop both autologous and off-the-shelf TCR therapies, with plans to expand into other solid tumors like lung, breast, and gastrointestinal cancers. The platform has transitioned from oncology to virology, targeting infectious diseases including hepatitis B. The FDA's recognition highlights the therapy's potential and validates Lion TCR's strategy of targeting HBV-related diseases with TCR T-cell therapy. Lyo6M004 is an autologous cell therapy engineered with mRNA to encode T-cell receptors specific to hepatitis B virus antigens. Early studies show it can reduce viral load and clear infected cells with a favorable safety profile. The therapy aims to treat over 290 million people globally and has demonstrated promising antiviral activity. This FDA approval marks the third major regulatory milestone for Lyo6M004, following Fast Track and Orphan Drug designations for HBV-related hepatocellular carcinoma. A new breath sensor developed by researchers at Penn State could transform diabetes diagnosis. Led by Huanyu Larry Cheng, the device analyzes acetone levels in exhaled breath to identify diabetes and prediabetes within minutes. Elevated acetone, a byproduct of fat metabolism, is a reliable biomarker when levels exceed 1.8 parts per million. Unlike traditional tests that involve invasive blood draws or sweat analysis, this sensor offers a non-invasive, quick alternative. Currently, the sensor requires breath collection in a bag to reduce environmental interference, but future versions aim for direct use under the nose or integration into masks. The device uses laser-induced graphene created by a CO2 laser on polyimide film, resulting in a porous material that improves gas sensing. A moisture barrier layer has been added to prevent water vapor interference, addressing humidity issues in breath analysis. The sensor combines graphene with zinc oxide to selectively detect acetone, avoiding interference from other gases. This technology, recently published in the Chemical Engineering Journal, could make diabetes screening more accessible and affordable, especially in settings lacking advanced lab facilities. Unlike existing methods that depend on induced sweating or blood tests, this device only requires a simple breath sample. Researchers plan to study how diet and exercise influence breath acetone levels, potentially broadening its use for general health monitoring. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.